This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on August 7, 2023. I'm Vincent Racaniello. You're listening to the podcast all about viruses. Joining me today from the state of Maryland, Kostya Chumakov. Welcome back.
Thank you. Thank you, Vincent.
And from Germany, Raul Andino. Raul Andino. Welcome back, Raul.
Hey, Vincent. Thank you.
Both of you have been on this program a couple of times in various places. We're scattered all over the world today. But the goal today is to talk about NOPV2. The N stands for novel, is that right?
Right.
It's a derivative of Albert Sabin's OPV serotype 2, which I, a few months ago on TWiV, called a failure. And both Raul and Kostya emailed me and said, no, it's not a failure. It's not fair. So I said, come on and talk about it. So here we are. We're going to talk about it and give it a broad discussion. So first, let's talk about why was NOPV2 designed to begin with? What was the problem with Sabin's original OPV2 that necessitated a redesign? Maybe Kostya, you could start by talking about that.
Sure. Yes. I mean, when Sabin strains were introduced at the turn of the 1950s, 1960s, I mean, at the time, there was not much attention paid to the side effects that this vaccine can produce. And only by the, I would say, mid-1980s, it became clear that these strains are not really as stable as we hoped for, even though reports were trickling in earlier. But then, by the time when the United States and Europe practically eliminated polio, there was a realization that the only few cases of paralytic polio that are observed actually are caused by vaccine-derived virus. At the time, it was called vaccine-associated paralytic polio. It was a virus, Sabin virus, that accumulated few mutations. The common belief was that because of these mutations, the paralytic cases occurred. But at the time, as I recall, until the turn of this millennium, the paradigm was that, okay, yes, these strains are mutated. They can paralyze the vaccine recipient. In some cases, close contacts, but it's not transmissible. And the word was that these strains cannot establish chains of transmission. Mm-hmm. And then not until, I think, 2000 or 2001, when there was an outbreak of paralytic polio caused by vaccine-derived polio virus of type 1, identified in Haiti and Dominican Republic. And it was a complete game changer because it was clear that these mutated strains can not only paralyze their vaccine recipients, but can restart circulation, and by any measure of phenotypic properties, pathogenicity, transmissibility, they're not different from wild strains. So it's a kind of a runaway vaccine strains. And type 2 was particularly bad in this respect. I think that more than 90% of all cases of vaccine-derived poliovirus cases were caused by type 2. And of course, the polio eradication campaign was facing a dilemma. I mean, what do you do? I mean, this vaccine was instrumental in the eradication of polio. And by the way, type 2 polio was eradicated first in 1999. So it's like almost quarter of a century ago. But still, using this vaccine strain, type 2 specifically, was unacceptable because of the large number of vaccine-derived poliovirus. Mm-hmm. And then at the time, there was an idea suggested that maybe we can improve based on our knowledge of molecular biology and the genetic structure and all the features of poliovirus. maybe we can rationally design the genome to prevent it from mutating, from reverting to virulence. And that's how it started.
I have to say that in 1981, David Baltimore and I published this paper showing a DNA copy of poliovirus is infectious. And the last paragraph we wrote, it may be possible to use this technology to make vaccine strains that do not revert to neurovirulence.
Wow.
Okay. So the 1981, I never got involved with doing any of that, but that's the seed of it, I think. You remember that, Raul?
Yeah, absolutely. I did read read that paper. So I read the paper. I also wanted to just add one thing that, I totally agree with what Kostya was saying, but it's important to make the point that the Sabin vaccine was incredibly effective. Mm-hmm. Because, since the eradication campaign or the global vaccination campaign was initiated, the number of cases of poliomyelitis was reduced 95%, 99%. So it was from 100,000 to, few hundred. And so that clearly argued that the vaccine works not only in very controlled places, but in any place in the world. Under any conditions, if you do a good campaign, you end up with a very effective way to protect against poliomyelitis. And of course, the genetic stability was a problem because when the wild-type virus, because of the vaccine, start to wind down and disappear from circulation, now when they start to circulate with the vaccine-derived virus. Mm. So that was very clear, as Kostya said, that if we would eradicate the disease, and if you want to eradicate the cause of the disease, the agent that caused the disease, then you will have to also consider that the agent that caused the disease comes from the vaccination. And here's a very important point. I think it's very glamorous to create new vaccines against emerging diseases. Everybody wants to do a vaccine against cancer, or against SARS. But I think it's more important from a public health perspective to improve on vaccines that are already very effective, but they are not perfect. And so I think that was the motivation. Mm-hmm. And I think Kostya and I, for 20 years and more, were on the side of the field saying we need to find ways to improve this if we want to complete this job. It's like any other medical intervention. There are some medical interventions that are good and they are very effective to prevent disease in people, but they are not perfect. And it's very important how you apply the medicine to make it better. And so I think at this point, this is the critical point. We have a better vaccine. We can discuss more about it. But still, it's not perfect. We need to apply it in a way that is going to make it safer.
That's interesting. You say apply it instead of further develop it.
Well, Vincent, I think it's Raul raised a very important point, and that's something that I also actually stress when I give lectures on vaccinology, that vaccines are not static. They evolve. Think about all vaccines that we know now. Smallpox vaccine, right? It was created in the 18th century. Over the years, it was transformed. Then it stopped, and then in the turn of the century, it was redesigned in a way that it's more safe. The same with rabies vaccine. Now, pertussis vaccine. All vaccines, from time to time, they need revisions based on the changing epidemiological situation, societal changes, economics, and all of this. So this polio vaccine is not an exception. This falls into the paradigm of vaccines being needing evolution.
Mm-hmm. Mm-hmm. Yeah, I think, and this we'll come back to this at the end. I think NOPVs could use some further test redesign, perhaps. But as Kostya, you pointed out in our TWiV, we won't be able to do any more research on poliovirus, so it's not going to be possible to make any modifications that might be needed, and this is a real problem.
Mm. Yeah. No, I mean, the other thing that perhaps, for many people, I mean, I know that both of you know very well the advantages of an oral live attenuated vaccine, but I just wanted to point it out for people that are listening to us, and maybe they don't realize why is OPV so effective. And there's two elements, in my opinion, that are very important. One is that the virus mimic an infection, a real infection. And we don't understand really what takes in creating immunity to a virus and in such an effective way. And we believe that once you get infected with polio, you are resistant for life. I mean, whether this is true or not it could be debated. But the point is that this virus clearly induced long-lasting immunity, first. Secondly, it induced immunity in a place where you need it, which is intestinal immunity, because that will not only prevent disease, but also prevent transmission. And the third point I want to make is this vaccine is more effective at the community level because it is spread to close contacts. So you vaccinate the baby, and other people in the household also get vaccinated at the same time. So you might get a boost, or you might get vaccinate people that are not being reached by the vaccination campaign. So this expand the efficiency of the vaccine, or how much vaccine reach people. And I think those are critically elements that make this vaccine so effective.
Yeah, Raul, I mean, can I expand on this point? Because I think you touched it, it's extremely important. Because when Albert Sabin developed this vaccine, he insisted that it can only be used in campaigns.
Right.
He realized that it's not fully stable. It's only when you immunize the entire population of susceptible children on the same day, or perhaps on the same week, only then you can avoid this unfortunate side effects of vaccine-derived poliovirus and so on. And sadly, I mean, it's not practical. I mean, for some countries, they still do it. For instance, Cuba, the country that first eradicated polio in 1962. They didn't have a single case since then. But they still use OPV in campaigns the way Sabin prescribed. But in countries such as United States, it's not practical. You cannot immunize the entire population on one day. So that's why it's used kind of in a piecemeal fashion, so that, one kid gets it now, another kid doesn't get it until, 3 months later. And of course, the pool of susceptible children is quite big. So that's why the virus that is being shed by the vaccine recipient can start circulation. If it was a campaign, it wouldn't have happened. And just like you said previously, I mean, maybe if we were using OPV in a way Sabin designed it to, maybe we would need the novel OPV.
Yeah, that's a good point, and that brings up, Raul, you said that the virus can spread within a family. Well, that's how you get VAP, vaccine-associated paralysis, and if you massively immunize the whole family, you wouldn't get that, right? Yeah. So that's
exactly right.
That's part of the problem there. Yeah. So, how did this modification of OPV2 begin? Is it that can you pinpoint a time when you said, let's do this, or did it just happen slowly?
Yeah, I mean, I think this is a very important question, Vincent, that you bring up. And I wanted to emphasize this point. It's only as a consequence of basic science research for many years on the entire polio community that this vaccine was possible. It's, really is a merit of the entire community, including yourself, with your paper in 1981. But it is true that we knew, as Kostya said, that there was one or two particular areas on the genome that often will accumulate mutations that will make the virus to regain the ability to invade the central nervous system. So polio is a neurotropic virus, and it causes disease because it goes to the central nervous system, around 1% of the cases in the wild-type virus. And so this particular element or mutations affected an RNA element that was in the 5' non-translated region of the virus. And I think in general, people believe that it has something to do with initiation of protein synthesis because it's within the element that recruit the ribosomes to the viral RNA. But the very intriguing thing was that this particular element is an RNA element. If you make it more stable from a physical point of view, the virus is attenuated. If you make it more less stable, also the virus doesn't replicate very well and it's attenuated. So the question was, can we maintain the stability of this RNA element in a way that mimics the original vaccine, that doesn't get to the central nervous system, but it cannot revert quickly, because all the mutations that we saw in original OPV was one single point mutation. We did an analysis of people that after, and not only us, but other people as well, show that after few weeks post-vaccination, a single point mutation in this element was sufficient to make the virus neurotropic and neurovirulent. So the question was, can you introduce more than one mutation, maintaining this structure in a way that is not neurotropic, but the structure cannot become neurovirulent. I mean, the structure cannot support neurovirulence if it doesn't go through more than one mutation. So the idea was to increase the number of mutations that the virus has to accumulate before it becomes neurovirulent. And so that was, I think, the original idea, because that was the element that determined neurovirulence in most of the cases. There was also mutation, Kostya knows more than me about this, but in the capsid proteins, in BP1 and maybe in BP3.
BP3, yes. Right. I mean, I think that Raul, mentioned the most important part of how this novel OPV was designed, and I think it probably determines 90% of its stability and so on. And it's based, of course, on the first discovery that was made by Phil Miner and Jeff Almond, I think, in the early 1980s, that demonstrated that this domain 5, as we call it, it's a stem and loop structure within the iris, internal ribosome entry site, that is determinant of neurovirulence, reversion, and attenuation in all three serotypes. It was remarkable that the same structure, even though it was different, mutations are different, but still the main motif was that if the physical stability, thermodynamic stability of this inheriting structure is greater, then the neurovirulence increases. If it is decreased, then the neurovirulence decreases. And this balance is important for maintaining the neurovirulence level. So I think that, as I recall, the first attempt to tinker with this element was done again by Jeff Almond and Andy McAdam, right? I think it was.
That's right.
Even way before this project started. So we knew that you can redesign it in a way that it will be more stable.
Correct.
But it's just one element of the whole design, because another element, of course, was the SCRE element. This cis-acting replicative element that resides in the middle of the molecule in 2C region, right? I think it was first discovered by Carla Kierkegaard, right? I'm sorry, Stan Lemon. I think Stan
Lemon discovered that.
Yeah, he discovered it. Carla, I think she discovered this high-fidelity mutant in polymerase, right? In Sweden. Which is a third element in this design, basically a mutation that makes polymerase more accurate. So it increases fidelity of the replication. So it's, as we call it, a hi-fi mutation.
I think,
and then the fourth element, of course, was what Raul discovered, a mutation that decreases the frequency of recombination events. And the fifth element of the design was not included in this particular design of this novel OPV, but was considered as a second alternative, is the something that Olenq and Kara Burns explored, the optimization of codon usage in the capsid protein, basically using codons that are not preferred by poliovirus. Poliovirus, for whatever reason, doesn't like certain codons, and if you put it in, all these codons increase their frequency, then of course the fitness of the virus decreases. So these were 5 elements that were kind of incorporated into the design to try to stabilize the genome and prevent it from accumulating, reverting to virulence.
So we have the 5' UTR. We have the Cre. The Cre element was moved from 2C up into the 5' UTR, correct?
Yes.
Yeah.
And then we have 2 amino acid changes in the RNA polymerase, correct?
Yeah.
Right.
And is there another?
So the idea is, if I can complete this, the idea was that, of course, the first stem loop that we call domain 5 was important and could be stabilized by introducing more than one mutation.
Right.
Problem was that the entire 5' UTR could be replaced by recombination with co-circulating enteroviruses. So to prevent that, we moved the Cre element. And so now if you replace the 5' UTR, you lose the good Cre element, which is next to this domain 5. And because you don't have the good Cre element in the 2C region, then the virus cannot replicate. So that recombinant was prevented by moving these 2 RNA elements together, and really works as a charm, at least in cell culture. And we never saw any recombination. I know we forced it to recombine. It was just incredible. We couldn't believe that this is was working so well. And then the mutations in the polymerase is an interesting thing because Carla Kierkegaard and we have both identified this mutation of high fidelity, and then we follow the high fidelity idea, and we show that the diversity of the virus population was important for virulence. And so in this study, we combine the high fidelity with the recombination, because recombination also is important to increase the diversity in the population, because the virus goes through a lot of bottlenecking, and if you don't have recombination, you cannot repair the genome. But with recombination, you repair genomes during this bottlenecking process. And this is inside of the host, of the infected host. So we put recombination and fidelity together in one polymerase. And we knew that either recombination or fidelity both will attenuate the virus, but together, now you have another 2 elements that independently will attenuate the virus, just by playing with diversity in the virus population. And then, as Kostya said, there was this third idea of codon de-optimization that was focused on the capsid protein.
Right.
And I mean, we can discuss why this one was not incorporated in the final. I think it was an issue of production. Kostya.
No, I think it turned out to be hyper-attenuated. Because, I mean, both candidates, candidate 1, the one that is now called novel OPV, and candidate 2, that included the same elements, and on top of that also codon de-optimized P1 region, capsid coding region. They were tested in clinical studies in a phase 1 clinical trial. It was a very small study, just 15 subjects, but the excretion of virus, as I recall, 1 or 2 subjects didn't get infected at all. So it was a little hyper-attenuated. So, and that it was kind of set aside as a kind of backup plan, and the clinical development continued with the first candidate strain.
Yeah, and I think that's an important point because, like I said at the beginning, since OPV is so effective in any population and any condition, you didn't want to change the biological characteristics of the vaccine by further attenuating, further reducing the ability of the virus to replicate. So the danger was that if you over attenuate the virus and make the virus less effective to replicate, now you will have some populations, some people that will not get effectively vaccinated. So we didn't want to change the replication fitness of the virus in the gut, but stabilize genetically.
I think over the many years I've thought about this. I like to use fitness now instead of attenuation because I think that's what it is, really. Yes. It's about reducing fitness, and if you can restore fitness, then it will somehow get into the CNS and cause neurological disease. So type 2 was chosen because there was a lot of vaccine associated type 2. It's circulating vaccine-derived type 2. But I wanted to ask you both, the original changes that McAdam and Almond and Miner introduced into stem loop 5, those were done on a type 3 polio backbone, right? And we know that type 2 OPV is the least fit of the three. So did you ever consider, we want the capsid protein of type 2, but did you ever consider putting the rest of the genome from type 3 just to mimic what McAdam and colleagues had done there?
Vincent, I want to correct you. Actually, type 2 is the most fit.
It's the most fit.
It's the most fit. That's why in the vaccine formulation, it is included in the lower concentration. And that is actually why it was eradicated first. Because type 2 is really a fit virus.
So then even with that correction, the question is still remaining. Why didn't you put it in a type 3 instead of a type 2?
Actually, it was done before, Vincent. If you remember, Akio Nomoto published a paper many years ago when he suggested type 1 as the backbone. And he created all three strains. It went unnoticed. I think it was in the late 1990s.
That's right, yes.
So, nobody thought about that we will ever need it. And by then, of course, Akio passed away and nobody was to advocate it. So I think that in retrospect, it should have been better. Because, look, there is a fifth element. Because all these four elements that were included, I would call them mechanistic. Because 5' UTR, it's a structure of the hairpin. It's a physical structure, right? You adjust mutations so that the physical structure will be as stable as it was with Sabin strain. Then a Cree element, it's also sort of a physical. You relocate something to prevent recombination. Mutations that are in the polymerase gene, again, they prevent, first of all, mutations. This hi-fi, they prevent emergence of mutants. By copying RNA molecule more accurate. And then mutation that Raul discovered, this REC mutation, recombination, it prevents for whatever reason, and Raul perhaps knows how this mutation works. But again, it's a mechanistic, event. But there is a fifth element that I personally was advocating at the time, but it was not really accepted. Not to change something that would change mechanistically replicative processes in virus, but rather to take away the pressure to recombine. Because if you think about, polio enteroviruses in general, they recombine, they're very promiscuous. When they get into the same cell, they exchange their parts, but they will not be selected unless there is a gain in fitness. And a few years ago, actually, I think maybe 6 or 7 years ago, we made a study with Vadim Agol. He provided us with a huge collection of vaccine-derived polioviruses. I think there was more than 120, I think. So we deep sequenced them all. And then compared what serotypes like to recombine, what parts of the genome they prefer. It turned out that type 2, if you have a recombinant type 2, it never, ever retains 3' end. There is a tremendous, dramatic fitness loss at this. So this 3' end, the second half of 3D gene, is so bad in type 2. So viruses wants to get rid of it any way it wants. Type 1 has a very bad 2A. So that's why it has to recombine. But it's not as bad as type 2. So, I mean, if you design virus in a way that you use P2 protein, central part of the genome from type 2, and then you use type 1 P3, then it would be the best, the fitness of this virus will be the highest. So that's why this virus, if you redesign it this way, there will be less pressure for virus to recombine. But I guess it was too late because, I mean, of course, there was a timeline, so there was no time to explore this possibility, and the type 2 based strains were already available, so that's how it happened.
Okay. All right. So after the phase 1 that you described, the Poliopolis study, right? What other trials were done before this was broadly used?
Well, then there was the study we did. There was a study in Panama, and it was actually it was a series of studies. Initially, was a study with. Correct me, Kostya, if I'm wrong, but I think it was toddlers initially. They were already previously vaccinated with IPV, so with the inactivated vaccine, to make it safer, and based on those results, and in terms of, I guess it was two things major. One, the vaccine didn't cause any problem with the kids, and the second thing was to make sure that this vaccine was more stable than previous vaccines. And then from that, it was moved to, I think it was almost 800, 900 individuals. In this case was newborns and also toddlers, but with no history of vaccination. And so that was a big trial. I mean, it just was an incredibly achievement. And I think that was the phase 2, and I don't think it was anything else in the form of phase 2.
No, I don't think there was formally there was a phase 3 trial.
No, not yet. Yeah. So I think that there is a plan for phase 3 coming up. I think this year or next year, early next year. In a much larger, I think this is the Bangladesh trial that is being planned.
Well, that would be a good idea, I think, because, I mean, in those trials that Raul described, the phase 1, of course, it was in adults, phase 2, and there's 2A, 2B, I mean, whatever, there are some regulatory distinctions there. I mean, there was no, safety signal. I mean, everything was perfectly fine. And in part, perhaps it was because it was conducted in countries where there's not much enterovirus circulation. So I think that the critical part would be to conduct a study in a place where, enteroviruses are very prevalent, so that there will be a higher chance for this vaccine to find a partner to recombine. And the third consideration is that it was used as a monovaccine, basically monovalent vaccine. Of course, OPV is given as a trivalent formulation, providing an excellent opportunities for viruses to exchange parts. So that's why I mean, this first clinical studies, they did not completely mimic the situation that the vaccine would face in real life.
So I want to ask you both, if vaccine-associated paralysis is 1 in 1.5 million doses, these trials were not big enough to know if you had improved on that, right? And so were you still comfortable having this now? It's been in over 600 million kids, I believe, in Africa. Were you okay with that decision?
Well, the issue was, it's important to point out that we knew that this number because of the use of the vaccine, and we also knew by sequencing some of the mutations that were associated with increase of fitness and increase of virulence. So one of the things that we did And this obviously was a major clinical undertaking, but we basic scientists were still working together with those teams to reconstruct some of these mutations observed in the field in the original vaccine. Mm-hmm. And then we tested in an animal model that it has been accepted by WHO and the World Health Organization and by many studies as a good model to examine neurovirulence. In fact, this model derived from your original PBR transgenic mice, Vincent, of course, but I think in a different background of mouse, which is more susceptible. So that study, this combination between clinical, re-engineering those mutations on the original NOPV and then put it back on the mouse, give us certain confidence that those mutations that were accumulated, observed, and the sequencing and the clinical trials were not increasing the neurovirulence to a level that was dangerous. Mm-hmm. So that gives confidence. So that's one issue. But of course, in all this business of vaccination is, how much is the risk and how much is the benefit. The risk is very high because there was a lot of epidemics going on derived from the vaccine that were not controlled by the original OPV. And this is when WHO decided to provide this emergency use authorization to the new vaccine. So there was enough evidence from the preclinical data, but also from this clinical data, that the vaccine was better. No, it couldn't be worse. It was better than OPV. So that was the mindset in WHO to say, okay, the evidence suggests it's better. We don't know for sure it's better, but it's not going to be worse than the OPV, original OPV. Mm-hmm. And so they gave this authorization, and the next few years, everything looks really good. There were several epidemics were completely stopped by use of the new NOPV, which was not the case of using the monovalent OPV2, the original OPV2. So it was very, I don't know.
Mm-hmm. Yeah, no, I think it's very important that Raul just mentioned, because this was a decision that was made from the very beginning, that, safety in terms of neurovirulent safety would not be a primary endpoint in clinical trials. Because, with the VAP incidence of 1 in a million, roughly, you would need 5 to 10 million subject study, which is completely, it's not feasible at all. It's one thing. Second the second reason why it was very difficult to assess comparatively safety, because at the time when this vaccine went into clinical trials, this novel OPV, OPV2 could not be used anymore because it was phased out. Trivalent OPV was replaced by bivalent vaccine that did not contain OPV2, OPV2 strains, and Sabin 2. In general, type 2 poliovirus had to be handled at BSL-3 conditions and so on. So it was not possible to conduct, side by side, one arm old OPV, one arm in the clinical trial with novel OPV. That's why the comparison was done with the historical studies that were conducted previously in a different place. So, doing it, accurately would not be possible. So the decision was made that we will use only non-inferiority, as they say, criterion, that this novel OPV should not be worse than OPV. And based on the preclinical studies, and we've done a lot of passaging of the virus and unfavorable conditions, 10 passages in a row, and looking at all the mutations by deep sequencing and this and that, and testing in transgenic mice. All this showed that this virus is more stable. So there was a confidence that we will not need this, basically superiority, proving that it is safer.
Okay. So now the NOPV2 has been in over 600 million kids, correct? And can you summarize what we have learned? There has been some vaccine-associated paralysis, correct?
As I said, in general, the news were very positive until very recently, January, February of 2023 or something, and that start to be in the Congo Republic, Democratic Republic of Congo, and
I think Burundi.
There was
also Burundi.
And Burundi, yeah, which is next door. And of course, what happened is there was 2 independent emergencies. So in DRC, and some of those viruses spread to the country next door, Burundi, and that clearly is bad news, but not completely unexpected, as many people have said. An improvement of the stability doesn't mean that is a completely impossible for the virus to evolve. I mean, as Kostya said, there are elements that makes these viruses to look for any kind of trick, recombination, mutations to improve fitness. And if the selection is strong enough and the right partner is there to recombine, this gain of fitness can occur.
And I think we even know how it happened. Because I think that these strains, these recombinant strains, derivatives of novel OPV that cause these cases in the Congo, they were sequenced. I think Javier Martin's group did this. And it more or less is clear how it happened. I think that the first recombination event added additional Cree element where it belonged in 3C. And perhaps 2C, I'm sorry. Probably driven by the low fitness of the type 2 3' end. And then this intermediate virus had 2 Cree elements. One where it should be in 2C, and one that was supposed to protect the 5' UTR from being recombined out. And then it was easy. This second recombination event could easily get rid of the 5' UTR, replace it with the wild type virulent structure, and here you go. But again, it takes 2 recombination events to get to that place. And as a result, the stability of, even in terms of neurovirulence, the stability of this novel OPV was remarkable. Because like you say, it was 600 million doses distributed of this vaccine, and just 1 or maybe 2 instances of this recombination event. It was calculated based on what we know about the stability of conventional Sabin 2 strain. There would be a few dozen such cases if it was just an old vaccine. So, of course, it's not absolutely stable, but the stability is perhaps, 50 times, and safety as a result. It's by orders of magnitude better than the conventional vaccine.
Perhaps one thing that now to bring up in the discussion is that in DRC in particular, the social and political situation is very unstable. There is a conflict. There's more than 1 million people displaced, living in refugee camps, which is a perfect place because the sanitary situation to bugs to start to spread through the population in these refugee camps. In fact, there have been epidemics of measles and cholera in those camps, and that's clear evidence that many things are going on. And now there are evidence that there's a lot of enteroviruses also circulating in the camp again, because like Kostya said before, if you give the right partner and the opportunity to the vaccine to recombine with a co-circulating enterovirus, the vaccine will recombine eventually, and it will regain fitness. And, of course, the other problem was, like we discussed at the beginning, there was not enough coverage because it's not safe for the vaccine, the agents that deliver the vaccine in these places to go and vaccinate people. And so I think, it's very sad that some people got sick out of this medical intervention, but this gives us the opportunity to understand better what are the conditions we need to have before we deliver the vaccine, because in certain conditions, this medical intervention may be dangerous. So I think this is give us some limit now. Okay, it's good up to here, but if we use it this way, it's not safe.
But you can't not immunize certain populations, right?
Of course.
So for example, would IPV be a reasonable alternative in that situation?
It could be, of course. I mean, if there is infrastructure in a country to deliver IPV,
sure.
and the cost is taken care of because it's more expensive, then of course, and there are people who advocate, like Jacob John in India, for instance, he's a strong advocate, and they've proven that in tropical countries, IPV works better than OPV in terms of efficacy. Because all oral vaccines in this tropical climates, they are not as effective. It also, rotavirus vaccine is the same thing, because there's a preponderance of a huge number of enteric pathogens and this, what they call tropical enteropathy, and so on and so forth. So yeah, IPV could be better. But I think that even OPV could, look, it was remarkable when Raul and Q first discovered this CVD-PV phenomenon. I mean, of course, there were reports trickling in about type 1, type 3, and this and that. But the explosion of type 2 CVD-PV started at the time when trivalent vaccine was replaced by bivalent vaccine. And it's clear why, because the population immunity to type 2 decreased dramatically. At the time, it was supposed to be mitigated by giving IPV, but there was not IPV for the whole world. So, of course, the immunity in some countries dropped below the certain level. And, of course, CVD-PV started emerging. And the sad thing is that 20 years ago, we published a paper, and I can quote from it. It says, OPV2 should never be used to control outbreaks. And it was based on an experiment that was conducted in the Soviet Union in 1963-66. At the time, they were using OPV, trivalent OPV in campaigns. And then at some point, they decided nobody knew about VAP or CVD-PV. It was believed that vaccine is fully stable and safe and so on. So they decided to test how long the immunity lasts, basically. And they, in one region of at the time, it was Soviet Republic of Belarusia. Now it's an independent state of Belarus. In Mogilev region, they stopped immunization at all from 1963 to 1966. So there was no vaccine used at the time. And then only 50 children in one, it was a kind of orphanage. Were given OPV. And all of a sudden, there was an explosion, a proliferation of vaccine-derived polioviruses. So it was described, nobody paid attention, but fortunately, Vadim Agol kept the strains. And then we sequenced them. And it turned out they were all independent lineages derived from Sabin 2. So it was remarkable. And it demonstrated that if OPV2 is used in a population that is not immune and it's given an opportunity to circulate, it will inevitably develop into something bad. So that's why the conclusion was to never do it. So unfortunately, it was not heard, and this switch from trivalent OPV to bivalent was done in a haste. At the time when nobody was ready, there was not enough IPV. And after this switch, the number of cases increased, I think, 1,000-fold. Before the switch, there was just 2 cases a year. At the height of this CVD-PV pandemic, I would say, it was more than 1,000. So, and it was only because the epidemiological situation was not taken into account.
So I guess my characterization of this as a failure was because of this paralysis and the idea. Raul, you published a paper in Cell Host Microbe, engineering the live attenuated polio vaccine to prevent reversion to virulence. So I'm kind of holding you to a high standard. That's why. And so you have a couple of kids in DRC and Burundi. The U.S. switched from OPV in 2000 because vaccine-associated paralysis was no longer acceptable, and it's still, I think it shouldn't be acceptable in any country, not just the U.S. And so we should continue to improve this vaccine or make a different one so that kids don't get paralyzed. That's my feeling. Yeah.
Okay. Two reactions to your question. One is the title of that paper. As you know, when you write a title, you can, you play around with words all the time, and it has to get into 150 characters, and so it's complicated. So to prevent is, the intention is to prevent it, right? But you cannot write the whole thing, right? It maybe is a little misleading, but if you read the paper, and both in the introduction and in the discussion, we clearly say this is an improvement. Whether it's completely prevented or not, we need to use it, and people you use it, and it clearly now it shows that there is very much rare cases in which you get this problem, this reversion that causes disease, and this is very sad, of course. And I totally agree with you. This is not acceptable to America or Europe. It's not acceptable to Africa either, right? That, I think, is a moral issue. Now the question is a moral issue in this sense. If you don't use it, and you say, okay, we're not going to use it, you end up in the case that Kostya was describing, right? So you stop vaccination, and then you start to have all this emergence. Then you say, well, let's use IPV. And this was tried, and there's two problems with that. First of all, IPV is more expensive to produce and to deliver. So it's impossible to imagine that if OPV cannot be delivered in DRC, IPV will not be able, because you need to have syringes and specialized medical personnel, etc. But the second issue that I think is very important is IPV will not prevent transmission, silent transmission of virulent viruses. And so the moment that you reduce your IPV, let's say you have enough IPV for a campaign, but then you reduce IPV, and the newborn babies, will be completely susceptible because the virus is circulating in the population, but in a silent mode. Nobody knows that the virus is there because there's no paralyzed people, but the newborns are not vaccinated with IPV and will be confronting with a circulating virus.
Yeah.
So I think from a moral perspective, also you can justify that you need to improve it. And so the question, I think it goes back to your original question, how do we improve this? And then the question is, you can improve it from a molecular biology, virology point of view, or you can improve it from a design of the vaccination campaign. And I think both arms has to be used. We need to design good vaccination campaigns to improve it with what we have right now, and learn from what we are learning now with the new OPV or the old OPV, and try to perhaps, I think it's very hard to, Kostya was, for the last 10 years, we've been working together and trying to produce this thing, right? And it's been such a difficult thing. And you can imagine people say, okay, let's put this other mutation. And we thought, let's add this other mutation, or let's not use type 2. But at the end, it's such a complicated business, develop a vaccine.
Yeah. It's much more complicated than most scientists who work in the lab imagine. It's a totally different ballgame. But Vincent, I want to bring yet another objection to your characterization of this as a failure, because the way novel OPV is being used now is the most unfavorable. Mm-hmm. It's being used as a monovalent vaccine to stop outbreaks, meaning that it is being used in a population of naive people, people who don't have immunity to type 2. So that's why the pressure for this virus to regain fitness is the highest. If it was used as a trivalent combination, and it was used throughout the world, if and when we get this trivalent vaccine, I think it would be the solution to the entire polio eradication game. Because, if you immunize everybody with this novel OPV of 3 serotypes, then every single child will get immunity for life. And then there will be no fertile ground for CVD-PVs to emerge. And since the vaccine is at least, 10 to 100 times more stable, there will be basically no problem. And before the switch to bivalent OPV, CVD-PV type 2 was not a problem at all. It was a man-made disaster. So I think the solution is just in front of us. We almost have this vaccine in our hands. Development of a new one is not possible because of many reasons. Inability to work with polio and enormous costs. Nobody will want to sponsor this effort, but this vaccine is already there. So I think that the next step should be to the development of the trivalent formulation and use it throughout the entire world.
So we should point out that NOPV 1 and 3 have been made, correct? It's being clinical trialed. What is that, as a bivalent formulation?
No, right now it's a monovalent formulation, and I think it's been now in phase 2 trial. So the next phase would be to develop trivalent formulation, which is not an easy task either, because you will need to balance 3 different products into one formulation so that they do not interfere with each other and still produce good immunity. That would be still work to do. But I think it is the best opportunity for us to get a new vaccine for post-eradication world, because it's clear we have to continue immunization indefinitely. Nobody should ever think that we can stop immunization. No country should be allowed to do this. It would be a kind of crime against humanity, because creating a population that is naive, doesn't have immunity, would be a kind of ticking time bomb. So at some point it will explode. So we must continue maintaining universal immunity of the entire world population forever. So, and this vaccine is perhaps the best tool to achieve this.
So when you say eradicating polio, you're talking about the disease poliomyelitis, right? Not the virus.
No, we will never eradicate virus. Virus is here to stay. We just have to learn to live with it, so it will not do us any harm. And the only way to do is to maintain universal immunity. That's it. And the reason why I think that this tool is better than IPV, because you could do the same with IPV. But IPV does not prevent virus spread. Basically, if you're immunized with IPV, you will never get paralyzed, but you can be infected and shed virus the same quantity as the person who was never immunized. So communities immunized with IPV can sustain transmission, silent transmission, no cases. No harm done, but virus can circulate. And since we live in a world where there's an increasing population of people who are not immune by different reasons, either refusal to immunization, some medical treatments that are immunosuppressive. So there will always be a certain small number of people in any population that would be vulnerable to this silent circulation. And then it can hit somebody, like this unfortunate guy in New York City last year. I think it was, right?
Last summer,
Last summer. Yeah, so there's always somebody who is vulnerable. So that's why a vaccine that creates a mucosal immunity is much better than the one that doesn't.
So let me ask you a question, and Raul. Scandinavian countries have used exclusively IPV, and they claim they have no polio and no circulating virus. Is that possible?
Yeah. I think, this is an interesting case. I think the transmission of virus in hygiene countries, like in the Scandinavian countries, is more respiratory or more, something on the. It's not so oral fecal like in the most of on the world, and so you can stop it perhaps by using IPV there, the circulation. I think it's not completely impossible. It's a little bit with the RNA vaccines for SARS. Those vaccines in general do not stop virus circulation, but it reduces a little bit of the virus load and it reduces the probability. So depending on how the virus is transmitted, that you may use IPV, but you're not going to be able to use IPV to stop circulation in places where the sewage is not available, or there's less conditions that prevent this oral fecal transmission.
I've heard Phil Miner and others say this respiratory transmission, but I really have never seen the data that would support that. Couldn't it be that there is actually circulation of poliovirus in Scandinavia? We just don't detect it. And if you get IPV, it will boost mucosal immunity if you've already had a poliovirus infection, right?
Yes, absolutely.
Look, I think that there was an example. I mean, of course, there is circulation in these countries, of course.
Okay.
And Raul mentioned that this force of transmission, whatever it is, kind of high hygiene or lower kind of moderate climate and so on. That's one part. Second part, of course, is that in these countries, the infrastructure for vaccine delivery and compliance, population is very kind of, they like to be immunized. There's not much vaccine hesitancy. But we have few cases in these countries. For instance, there was an outbreak small outbreak in a kind of religious community in the Netherlands. It's kind of people who do not immunize, but they got the virus anyway, because there is a circulation in the Netherlands. In Finland, in the 1980s, Finland also never used OPV, but their neighbors, the Soviet Union, used. And there was an outbreak of type 3 poliovirus in Finland. We know that there is a circulation in these countries. And by the way, this oral transmission, this is something that Jacob John keeps publishing papers. He thinks that this is the only way polio, transmits, oral. He said there is no evidence. I disagree, respectfully, but there is such an opinion. And by the way, there were old papers showing that IPV causes some mucosal immunity of oral infection. Not to enteric, but I guess it depends on the country where the oral or fecal oral prevails in different places. But it could be that IPV causes some protection, but not as good as OPV.
So as recently as February, CDC has said there's still poliovirus in wastewater in New York. Okay, so people are still presumably OPV2 like viruses. And so the CDC has suggested that the U.S. should stockpile NOPV2 and perhaps use it to control this. They call this an outbreak in New York. So I'm wondering what your position would be on the U.S. Of course, they would have to get approval to use it, I suppose. But do you think that's a good idea, Kostya?
Well, look, in theory it is, why not? But I don't see how it can be done if people in some places in this county where this person lived who got infected, I think that the vaccine coverage is so low. For whatever reason, the population there is so reluctant to get vaccinated by any vaccine. So I don't know how CDC will go door to door. How this vaccine will be delivered, because those people who are vulnerable, the same people who refuse to get vaccinated.
Well, if you remember, they did a campaign last summer, and they delivered very few vaccines in the area. Nobody wanted to get to take them. So I think that's a really good point. But even more to the point is that this country, the U.S., has such a high anti-vaccine stance at the moment that if there is one case of paralysis caused by NOPV2, that's terrible for vaccination in general, I think. So what do you think, Raul? Do you think the U.S. should use it?
No, but I mean, I think the point. I mean, what we're discussing right now is critical also for vaccine development, because if you have a vaccine that is 100% effective, but only 20% of the population use it, it's 20% effective.
That's right.
So the question is, perception in the context of vaccine development is critical, and I would argue that the NOPV2 and NOPVs in general, 1, 2, and 3, are safer than the original OPVs. And we know OPV was safe for many years, although in certain conditions it's not safe. So I would argue that this vaccine is safer. The perception that this vaccine is safer could be a very nice element to increase the acceptance of vaccination against polio using this new vaccine. So, when we scientists discuss this, we always wanted to see what's the problem. And I'm totally open to this discussion. I think it's incredibly enriching and important, this discussion. For the general population, it's important to emphasize that this vaccine is indeed much better than the previous one. But all the evidence, all the facts...
Is what?
we lost Raul. Raul, we lost you for 5 seconds.
Yeah, you were about to say this vaccine, for the general public, it's important for them to know that this vaccine is, and then you got cut off.
Okay, that this vaccine is there's some interference. No, this vaccine is safer than the previous one. And that perception and that idea might encourage people to use this vaccine. And this is good for the entire population. So I think in general, it's important to convey the idea that we know the facts that we have accumulated in preclinical and clinical trials demonstrate that these vaccines are better. And they are, a lot better.
So question, go ahead.
I want to just finish Raul's thought. I mean, we should remember that at this point, countries that use OPV have a total population of 6 billion people.
Yeah.
So, I mean, switching all these countries to use of IPV would be a monumental undertaking.
Sure.
Replacing with this novel OPV will just mean nothing. It will be a positive public relations, aspect, saying that this is a better, improved vaccine. But the same 2 drops on the tongue of a child, and that's it. No needles, no pain, no crying. So I think that it would be much easier. And the cost, of course, the cost of this vaccine is probably the same as the OPV. So I think it would be a solution.
So I wanted to say that if, NOPV2 were to be authorized in some way in the U.S., it would just be to try and eliminate the circulation of type 2 poliovirus in New York, right? I don't think it would be routine immunization for everyone, but it makes me wonder where else in the U.S. is OPV2 circulating, right?
That's a good question.
We don't know,
and I think we don't know because we don't look at. I would bet that if you look in other places, it's going to be there.
Yeah, I would agree. So it doesn't make sense to just do it in one place because you've looked there, right? Which is New York.
Vincent, I don't think that U.S., Europe, and all countries of what they call developed countries will ever use novel OPV. And there's no need for it because, vaccine IPV is delivered as a combination, mostly as a pentavalent vaccine. Yeah. Taking it out will take a tremendous regulatory, undertaking. So, thinking that we can replace IPV with OPV, even novel OPV, I think it's a non-starter. Okay. But if those countries that still use OPV will switch to the novel one, that will be a solution. That there will be no source for this, CVDPV coming to this country or European countries. So that will be the end of the story. But some countries will continue with IPV. Some with novel OPV, at some point when all countries will be developed, right? I mean, they will all use IPV, but only at the time when they're ready, when their healthcare infrastructure, vaccine delivery, logistics taken care of. Then, of course, they can switch to hexavalent vaccine that would include IPV and diphtheria, tetanus, or whatever. I mean, all the cocktails of antigens.
But I would say, if I may add one point to this, because you asked me whether I would, bring NOPV2 into the United States. And I think because moral standards, if we use NOPV in Africa, why not to use it in America? I mean, from my perspective, I think the vaccine is safe enough to use it here. Now, you could argue that this will be, from a regulatory point of view and from the political point of view, very complicated. So what I would say is anybody because most of the cases of polio probably are importation from places where the virus is circulating. And so you could argue that this new vaccine that is improved should be incorporated as a one condition for people that travel to the United States to prevent the importation. But I think this is an interesting, it's way below my pay line here of what I can decide. But I would say that there is ways to use this new vaccine in a way that will generate more confidence that this is a good medical intervention that should be undertaken.
But do you think that people who receive NOPV, for the moment NOPV2, because that's what we have, do you think that virus will circulate less than OPV has circulated? Right?
I think, that's a very good question, and I think that it will circulate less in the sense that it's less fit. I mean, there is some reason why the virus actually reverts in the intestine. It is gaining fitness. There is something that the virus is not good about it. The vaccine virus is not good about it. So I think in general, it replicates well in the vaccinated people, but my census is going to not spread in the same way that BDVPs, the ones that actually regain fitness.
So it would be useful if you could get these viruses, the NOPV2 derived recombinants, and compare, at least in the lab, their fitness, right?
Yeah.
I think it's been done. I think that there are data from the clinical studies that suggest that novel OPV is less fit than the Sabin IPV, which is a good thing.
But I mean the recombinants that have been isolated from some of these kids that are paralyzed. I would like to know how fit they are, because if they are as fit as CDVP OPV2, then they're going to circulate as much, right?
Mm-hmm. Mm-hmm.
And I think that they're doing it as we speak. I think that they're testing it in mice and so on. So it will be done.
Vincent, one of the things that CDC and WHO are doing is when they conduct these campaigns, they not only collect samples from vaccinees and contact people that are close to the vaccinees, but also environmental samples. Mm-hmm. So, with time, we will know whether the NOPV2 lasts in the environment, how long it will last. And it will give us also a sense of,
I think that's really important. I have a sense that we're not getting the data as quickly as we used to. And I don't know if that's a real thing or if it's just my impatience, but I think it's been a long time since these kids, I'm sure the sequences are done and we should have them. I don't know why they're not being released. Do you have the same reservations?
Well, I think that publications take time. I've seen presentations at the meetings where they, discuss this data, but they will publish it at some point. But I want to stress one more thing.
Sure.
Vincent, you mentioned, will it circulate as well as the Sabin original virus? I think not. Again, it is a matter of population immunity. If this vaccine will be given to everybody, if everybody will be immunized, if there will be no gaps in immunity to type 2 virus, there will be no problem.
It's a big if, though. It's a big if.
Look, again, it depends on if you use vaccine or you don't use vaccine. So like let's say, is it a vaccine failure or failure to vaccinate? I think it's the latter.
I think that's a great point. And as you have already said today, we didn't use OPV2 properly, and that's what got us into this situation. So the assumption is we're going to use NOPV2 properly, and I'm not so sure about that. We'll see.
Correct.
But there was one thing I wanted to say, Raul. You mentioned that, we should, if we can use NOPV2 in Africa, we should use it in the U.S. Well, as you know, OPV, Sabin OPV, is no longer licensed in the U.S. because it's not acceptable to have kids paralyzed by it. And so it seems to me that it's a double standard that we don't license something here, but you can use it. For many years, it was used in other countries. So this is the other issue that I have. And I realize that your point was a good one, that you have to use something, and this is better, and so forth. But that's my other moral issue with this. Yeah,
look, some people make the opposite argument. They say, why United States and Europe, they use IPV, and they still have us use OPV. Why can't we afford it? Yeah. And that's economical question. When these countries are, strong enough financially to afford IPV, okay, but that will be their choice. I think that our, obligation as a scientific community is to provide them a cheaper option to protect their population, and that I think this novel OPV is an excellent tool for this.
So Kostya, you're familiar with production issues. Stan Plotkin has always said it's just economy of scale for OPV, and if you made as much IPV, it would be cheap. Is that correct?
Not entirely. I think definitely, producing large quantities decreases the cost. Second aspect, of course, is making IPV in countries kind of locally, like in India or China and so on. So that also decreases the cost, but it probably will never get as low as OPV. For the simple reason that you need a lot more antigen. You need roughly, I think, 10 times more virus particle for inactivated vaccine. And then, of course, there are steps to purify the virus, to inactivate the virus, to test. The production scheme is by far more complicated than OPV.
And you don't have any confidence in, say, empty capsid poliovirus vaccines as being viable, do you?
Well, look, personally, I think it's a great idea. It's unfortunately, so far, it's not, there yet. They're working on it. And the question is whether there will be interest on the part of, funding authorities to continue with this project. So far, it's been underwritten by the Gates Foundation. And I think that just like this novel OPV, I think we should be grateful to the Bill and Melinda Gates Foundation for doing this. Without them, it would not be possible. They also supporting
I think
this
vaccine-like particles, are actually very interesting approach. Particularly if you can imagine ways to also generate mucosal immunity using this vaccine particles. And I think that's the new frontier of vaccinology for sure. But the other thing that I think is important for people to realize that there's two things that you need to define in a vaccine. One is the antigen. The antigen has to mimic as much as possible the virus, but also the adjuvant. You need to recruit the right type of cells that will generate the long-lasting immunity. And I don't think we, at the moment, understand very well how to do that. So, it's trial and error. And in general, the vaccines that are inactivated are less effective than the live attenuated. So I think there's a lot more work to do to try to understand these basic aspects.
Yeah, no, this is absolutely right. I think that there are many good ideas and there are many new designs of vaccine that can be delivered orally. and induce mucosal immunity. The question is whether we will be able to continue working on these vaccines, because this restrictions on the work with poliovirus possibly chased most poliovirologists out of the field. There is a joke about a doctor and a patient. I don't know if you heard it. Like a doctor was treating a patient and prescribing this medication, that medication, and calling every morning, asking how he's doing, and then try letting some blood or do this and that. And then finally he calls and they said, he passed away. And he said, oh, what a pity. I had so many good ideas. So this is exactly what we're in now. Where the community of virology, we have so many good ideas, but nobody lets us do it.
Well, Kostya, you said in our TWiV that the people making the decisions that polio research have to stop are not people who know the biology and realize they think that there's no need for any other vaccines and that we're fine the way we are. And you thought that was a problem.
I think that they're approaching it from a very simplistic, point of view. They think that if we stop circulation of poliovirus, that's it. We can go home, take probably a prize or two, or get on the front page of New York Times or something, get some credit and go home. It's not going to happen. It doesn't work with viruses. Viruses are much smarter than us. They find a way to survive. They lived before us and they will live after us. So I think we have to learn how to protect from viruses. And in my mind, polio eradication is when nobody or very few people get paralyzed by poliovirus. That's my definition of eradication.
You do not feel that we can ever stop poliovirus circulation, correct?
I don't. And if we can do it, we will not be able to prove it. Because, like Raul mentioned, there is such a phenomenon of silent circulation.
Sure,
and it can take years before we discover it. So how can you make sure that there's no virus? Okay, it circulates. I'm fine with that as long as everybody is protected. Let it circulate. Who cares?
So immunization needs to continue indefinitely, basically.
Indefinitely.
Yeah, particularly if you don't have cases associated with the vaccine in any form.
Yeah.
Then you have a very safe vaccine, and you can continue with trivalent NOPV for years. Why to stop? Right? There's no reason to stop. In one day we'll realize that the virus is no longer the environment, and then fine, we'll stop it. But I don't think that it was actually sort of a motivation for people to engage on the eradication campaign, because it's actually very glamorous to say we eradicate not only the disease but also the agent that caused the disease, so the disease will never be seen again. Right, and we've been claiming for many years that the important role is to protect people from paralysis. So the picture of everybody in a Trump picture will have to wait for a few years.
So Raul, do you agree with Kostya that we cannot at the moment stop circulation of poliovirus?
I think so. It's hard to predict this because we don't know how the virus will react, and we don't know how the campaigns will be conducted. But I think if you conduct a very good campaign, and in an ideal world, everybody vaccinated, it's possible that you will reduce things and the virus will extinguish. I'm sure that many viruses extinguish over the course of history because the host that it was susceptible is no longer there. Whether it's realistic or not, very difficult to imagine. But I wanted to say one more thing. In all this business of eradicating polio, many people might be asking themselves, well, there is only 1,000 cases or 100 cases, 200 cases of paralysis in the world. This is not such a big deal. But I think what is a big deal is that it's the first time that we are trying, smallpox was the other example, but in this case, we're trying to do something that is, really difficult, and there is, many different levels of complexity from policy, regulation, science, and all these things has to come together. And if we can succeed and move forward to the next level, improve this system, this will spread to other diseases. We will know how to approach other problems. And I think the lessons learned on this eradication campaign can be applied to other things. And I think this is a very positive sort of byproduct of the reaction.
And Vincent, if I may, I want to qualify my answer whether we can eradicate virus. If at some point I started, collecting this data on the incidence, on the cases of polio. And if you plot it, it's kind of a line that goes down, and then now it's at a very, very close to the x-axis, but still lingering. If you plot it in a logarithmic scale, which it should be, because it's a chain reaction with, as epidemiologists say, R0 value less than 1. It's basically on the way to be extinct. So it's a remarkably straight line over the last 30 years, 40 years, way before eradication started. It's as a straight line, but it intersects the x-axis in 20 years from now. So what we're
this
dealing with...
This is cases of polio.
Cases of polio, worldwide cases of polio. What happens is actually the transmission of polio is slowing down year by year. I mean, of course, flukes back and forth, but still the are not effective reproductive number, as they say in epidemiology, is less than 1. So at some point it will get extinct. And it's just a matter of continuing our policies on immunization, as many people as possible, and it will be there, but it will take 20 years. Okay, that's fine.
Right.
Well, I mean, but many countries will continue to use IPV where the virus can be circulating, and I'm not sure why we're not really looking enough at environmental virus to be able to say it's going to be hitting the x-axis. I mean, disease I agree with, but I think there's more happening than we know because we just don't look enough.
That's exactly right. But I think that the practical conclusion from this is that, eradicating the virus should not be the end point for the campaign. It should not be something that we measure our success with.
I agree with that. Do you agree with that, Raul?
Yeah, absolutely.
Okay, so we can end this on an agreement then.
We pretty much agree on all what we were saying. I mean, there may be different the solutions may be slightly different what we have in our mind.
It remains to be seen if WHO and the people who run the campaign will agree with us. That's more important than three of us agreeing.
That's right.
Yeah. But we need everybody. We need WHO, and also, and we need a dialogue. We need to, learn from each other and try to improve this.
They're doing a tremendous job. But I think that what happens, I mean, the campaign has been 35 years. 35 years we've been pursuing the same strategy. I think that we'll learn so much about poliovirus biology, epidemiology, and everything else in this 35 years, but the objectives and the criteria of the campaign have not changed. We're still doing the same thing as if we don't know about CVD-PVs, as we don't have a new vaccine. I mean, it's still kind of running its course, and it's time for some correction, I think.
All right, that was a good discussion. That's a special episode of TWiV. You can find the show notes, microbe.tv/TWiV. If you have any questions, send them to twiv@microbe.tv. If you enjoy our work, we'd love your support. Microbe.tv/contribute. My guest today, Kostya Chumakov. Thank you so much, Kostya. Good to see you again.
Yeah, thank you, Vincent, for having us.
And Raul Andino. Thank you, Raul.
Thank you, Vincent. Always a pleasure to talk to you, to see you. Kostya as well.
Missed you at ASV this year.
Yeah, I know. I was traveling.
Yeah. When are you coming back to the U.S.?
The end of the month.
All right. Safe travels.
Thank you.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.